Good afternoon, everybody. Welcome to lecture. Hope you all had good weekends. We're going to start today with a molecule of the day. So today's molecule of the day is alpha tocopherol. This is also sometimes called vitamin E, and a lot of times it's referred to as the fat-soluble antioxidant, and there's certainly others, but it's one that a lot of times people think of when they think of fat-soluble antioxidants. We're going to talk about what makes it an antioxidant today. It has the ability to sequester reactive radicals, and that's really the key. And it all boils down to this particular OH group here on the far left side of the molecule. We're going to learn about that later today in lecture. The reason it's fat soluble is it has a lot of nonpolar character. It has this very long hydrocarbon tail here, and then a lot of the middle part of the molecule is also nonpolar. So it dissolves in fat well. That's in contrast to ascorbic acid or vitamin C. That's oftentimes referred to as the water-soluble antioxidant. So it's good to have different types. And this one is an important molecule and an important nutrient. So it's our molecule of the day. Any questions about the molecule of the day? All right, then let's start talking, or let's talk about the survey question from today. So survey question was one that gave you this molecule, which is a fatty acid. So I think we had a bit of a discussion about fatty acids last semester. So the fats that are inside plants and animals and us are made up of esters of fatty acids, and the fatty acid tails on these things dictates their function and what they're useful for. Some are used as energy sources, and some are used as starting materials for making biomolecules. And nutritionally, you need lots of different kinds of fatty acids. This is one real common unsaturated fatty acid. It has the acid part here, carboxylic acid, and a long nonpolar hydrocarbon tail, but it has a couple of different unsaturations in it. So this would be a polyunsaturated fatty acid, one that's got a couple of different double bonds in it. And it's called linoleic acid, and it's got a bunch of different points that are being indicated by arrows and letters. And the question wants to know, it says linoleic acid is a polyunsaturated fatty acid commonly found in plant oils. Which location would produce the most stable radical when a hydrogen radical is abstracted from the indicated carbon? So you got to think about what kind of dot would you get if you plucked an H dot off of each of these positions? And there are a bunch of different options here. So stability-wise, what do we know about radicals? What are some general trends from thinking back from when we were discussing these things? What makes a radical stable? Well, substitution was one thing. If you put your radical on a more substituted carbon, it's better. And then the other thing that really had a huge impact was stabilization by resonance. Yeah, so if you can make a resonance stabilized radical, that really makes a huge difference in terms of stability. There are a number of different places that are not resonance stabilized. Well, a couple. E is not a resonance stabilized position. If you put a dot on that end carbon, it's not resonance stabilized. So this one position there doesn't benefit from any resonance stabilization, and it's a primary position, so that's also a really not a very good place to put a radical in terms of stability. There is also an arrow pointing to the carbon of the double bond directly. Now that is not an allylic position. We just learned about allylic positions last time. It's the spot next to the double bond. This is the spot that actually is one of the carbons of the double bond. And if you put a dot there, it turns out that's not resonance stabilized, and it's not very stable at all. It's actually pretty unstable. So that's not a good spot. But then you've got three positions that are in allylic spots. You've got the A position, which is a sp3 hybridized carbon that's next to this double bond. You've got C, which is a allylic position that's next to two double bonds. And then you've got this allylic position D that's next to the upper double bond. So we've got three spots where if we plucked an H dot off, we would make a radical that's allylic and resonance stabilized. The difference is that the C position is adjacent to two different double bonds, and so there's more resonance structures you can draw. If you put a C dot there, you can delocalize the radical to put the radical onto this position. if you put it there. You can also delocalize it to put the dot out on that position. So there's more resonance structures that you can draw if you have the radical between the two double bonds than if you have the radical on either one of the ends here. So it's just a matter of how delocalized is the dot. The more delocalized it is, the more resonance structures you can draw, the more stable it is. So this concept isn't one that we hadn't talked about it in class, but I was just putting this out there to see what people would do with it, if you could kind of reason through what might be the right answer. Now we're kind of covering it in some detail. That position is sometimes called doubly allylic because it's an allylic position in two different ways, and it produces more resonance structures. So in terms of reactivity, that spot right there is really reactive, and it is a bit of an issue with fatty acids like this that are found in foodstuffs. So fats that are unsaturated and are in foods are subject to oxidation. In other words, they can react with oxygen, and the places they do it are at these really delicate allylic positions, those really reactive spots. So oxygen can pluck an H dot off of these allylic positions pretty easily, but this middle spot between the two is particularly susceptible to oxygen attack. And then the corresponding oxidation products produce rancidity. The fat tastes bad and you don't want to eat it anymore. So a lot of times foods contain antioxidants, and the purpose of those antioxidants is to try to slow down and prevent that oxidation reaction, the plucking off of H dots off of unsaturated fatty acids. So that's a bit of a discussion about today's survey question, and the correct answer was C. But if you answered A or D, give yourself a pat on the back because those are pretty close. So let's see how the class did. C was really quite popular, and then I think people were just confused by this B. They weren't sure what to make of it, but now you know that position is called vanillic. It's not allylic. It's when it's actually on the double bond. That's not good. So next to the double bond is where you get the stability due to resonance. So and C was a very popular answer, B was a popular answer, and the others were pretty minor. So I would say overall pretty good, and those that suggested B, just recalibrate and then you'll be good. So any questions about this survey? All right. By the way, I asked ChatGPT about this question. I took a picture of it and I asked you answer this question. I didn't give it any hints or anything. It did a pretty good job. It selected C. In its explanation, it made a couple of errors. It confused that spot as an allylic position, and then I called it out on that. And then it said, oh yeah, that's a vanillic position. And I said, no, it's an alkyl position. And then it said, oh yeah. And basically it kind of went around in circles. And so it basically got the right answer, got the explanation mostly right, but there was a little something wrong with it. And I had to kind of coach it to get it to the right answer, the complete right description. So, again, pretty good, possibly useful as a study tool, but, not without little quirks and errors sometimes. So, okay. Let's talk about some radical chemistry. We've been discussing this radical stuff for a while now. We're going to mostly finish up the radical chapter today. So we were talking last time about allylic bromination, and I want to talk about that some more, and then we'll go to antioxidants, and then we're going to talk about polymers. So this is going to be an exciting lecture. We've got a lot of really interesting stuff coming your way. Polymers, I think, is particularly interesting because that's forms the basis of most of the plastics that we use in our modern sort of society with packaging and products that are made out of plastics. A lot of them are formed by radical polymerization reactions. So radical reactions are used frequently to make these plastics. And so when we get to that unit, then we'll have a big explanation of where high-density polyethylene comes from, and polystyrene, and polypropylene, and all those other molecules that are kind of like it. So, allylic bromination. We talked last time that this is a reaction that occurs at the position next to a double bond. Just like in our survey question, we were talking about those positions as being special. Here's an example of a molecule that, if you were asked to determine what the allylic bromination products would be, it would be actually pretty challenging. So you might see, and this is the most complicated example that we're going to go through. It's kind of a worst case scenario in terms of like, what's the most complicated question you might get on a homework set or on an exam. This is going to be it right here, just because there's a lot of different products and a lot of things to think about. So if you saw a molecule that contains a double bond, carbon-carbon double bond, and you saw below it or above it, somewhere over the arrow, around the arrow, this molecule, this is N-bromosuccinimide. You might see it abbreviated NBS. I would also give you the structure on an exam, but if you're looking at a homework problem in ALEX, they might just throw you NBS. And then there's usually some activation energy like heat or light energy or heat energy. And then you might be asked to produce all the allylic radical products. So what do you do? Well, it's a stepwise process, I would say. And what I would do if I was solving these things or trying to solve these things is, first thing, figure out where all the allylic positions are that have hydrogens on them. So let's do that. We've got our carbons of our carbon-carbon double bond here. Look next door at all the positions that are allylic. So we've got H's here that are allylic. This is an allylic position. That's CH3. There's a carbon here that has H's on it. That's an allylic position. It's next to the double bond. The other carbon of the double bond, there's an H on one of it, but there's a C here that's got a couple of H's on it. These are allylic as well. So we've got 3 different allylic positions, and each one of those can react in this allylic bromination reaction. So what I would recommend doing first is predict all the allylic radicals that could form. Because the products that you get come from radical intermediates that are going to be at the allylic position. So I would say to work through this problem, let's figure out what are the radicals that we could get if we plucked an H dot off of this position or that position. Let's just draw all those out. And I'm going to do that over here. So we can handle them from in any order we want, but we can go from like let's go from left to right in this molecule or clockwise. The first one that we would get would be a dot in this position right there. And there's still one H attached there that I haven't shown, but this is one allylic radical. It's a dot that's next to a double bond. If you ever find yourself doing an allylic bromination reaction and you have a dot that's not next to a double bond, double check that because it's going to be wrong. So it's always got to be a dot next to a double bond. So there's one. What's another one that we could get? We could get one where we pluck an H dot off of the methyl group, and then we'd end up with that radical. That's if that one of those H's got plucked off in the reaction. And then finally, the position down here in the lower right corner, if that spot gets abstracted, H dot gets abstracted off that carbon in that position, we would end up with a dot here. Okay, now if you just were to imagine bromination products that came from those radicals, you'd end up with probably about half of the products. So that'd be pretty good. Just replacing the dot with a BR, because the next thing that happens is the carbon dot, like say for example right here, grabs an H dot off of an N-bromosuccinimide molecule. But to really answer this one completely, you need to consider the resonance structures of all of these species. So what we're going to do now is draw resonance structures of each of these 3 radicals, because they all are resonance stabilized and delocalized radicals. So we got to figure out what the other carbon is that contains radical characters. So we're going to do that next. So I'm going to draw a resonance structure for this one. And the way we can push arrows around here is if you start an arrow at the dot and put a half-headed arrow, a fishhook style arrow, to push it in, to share it between these two carbons and then grab one of the H's out of the pi bond and push it that way, that's saying make a new carbon-carbon double bond here with that dot and one of the electrons in that bond. And then the other electron in that pi bond has to go there. So with that set of arrows, you get a resonance structure that looks like this. Now you have a dot right there. So these two resonance structures represent one of the three possible allylic radicals. You can do the same thing with this one here. With this radical, we're going to take this dot and share it differently. I'm going to pair it up with one of the electrons from that pi bond, and the other electron in that pi bond I'm going to give to the carbon over there. And then that generates another new resonance structure that looks like this, where we've got double bond here and a dot right there. Okay, and then we got one more to worry about here. That's this one. If we think about the resonance structure for this lower radical, I'm going to take that dot and push it in, share it differently by pairing it up with one of the electrons in the pi bond, and the other electron in the pi bond I'm going to give to that carbon right there. Now we've got this other radical that looks like this. So that's a collection of three different allylic radicals and all their resonance structures. So if you get this far, basically you've done kind of the majority of the work in solving this question. What you need to do next is just realize that each one of these dots can pick up a bromine and become an allylic bromide product. So once you got your collection of resonance structures, the next step is just carbon dot gets a Br dot and becomes a bromide. So you can just basically recopy all of these structures and just change the dot to a Br. So once, yes?
Would we have to specify stereochemistry?
Good question. Yeah, you know, there is stereochemistry with these things. When you put in a bromine, you're going to make a new stereogenic center in many of the reactions. I think actually all of them except for this one. So yeah, it gets to be complicated, and it gets to be complicated enough that we're just going to not worry about stereochemistry for these. And that's just by tradition. I mean, when you look at these problems in all the organic textbooks that are out there, they just ask you for constitutional isomer products, just to keep it simpler. So the point of this question isn't to test your stereochemistry, it's to test your ability to generate resonance structures and rationalize through products that can result from them, but without worrying about stereochemistry. So it's a really good question. And we will just not worry about stereochemistry. And in order to talk about that in words, the way you would see this worded would be something like, draw the constitutional isomer products that would come out of this. Constitutional isomer allylic bromide products. Someday I'm going to come in here and screw this board to the wall. It just bugs me. But I left my toolkit at home. I've got a pretty good set of tools. I could totally do it, but anyway. Just like a big hammer and a nail and just like stick it through. Okay, well, let's just imagine what happens. I'm going to redraw these over here. And so this is just a recopying exercise at this point. So these are the upper two structures. Copied from here and here over to here and here. And then I'll take these two structures and recopy them. So I'm not doing anything new here. I'm just copying them verbatim the way they appear over there. And then we'll do a little bit of light editing to make them into bromides. So if you're working through these on like an Alex homework set or something, or on a practice exam, I would definitely have a piece of scratch paper where you can write the intermediates out. It's super helpful to have that. Just so you know, because you can't keep track of all this in your head. There's just no way. I mean, it's pretty easy to look at it on the board. I don't think it's overly complicated, and just that there's a lot of things going around. But if you keep track of them by writing them down, I think it becomes a lot more manageable. All right, there are our collection of 6 allylic bromide and radical structures. So what to do now? Just convert the dots into BRs. So I'm just going to erase the dot in each case and draw a BR. Dot, get rid of that, put a BR. Put it over here, remove the dot, add a BR in its place. From here, I'll take that, put a BR there, put a BR here. Mechanistically, what's happening is the carbon dot's grabbing a BR dot. We're not going to show that with arrows because it would, we could spend pretty much the entire lecture drawing mechanisms for every single one of these steps. We don't really need to, I don't think. I think we just need to know what's happening and have a quick way to predict products. So there you go. That is the collection of products without stereochemistry. There's stereogenic centers here, 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 and there, but we're not specifying because we don't want to, we don't want to like have 10 structures on the board. Well, we already got 6. So okay, but there is one other thing to worry about with these reactions. Sometimes the products that get produced, you get duplicates sometimes. So with some of these, with one of these, there's a set that's a duplicate set. In other words, yeah, and if you look, it's these bottom 2, because the 2 radicals that they came from are just rotations of each other. The products that form the constitutional isomers of each other are also the same. So these are the same. So this is one where there's, after you get through doing all of this stuff, there's an additional wrinkle, and that's that draw all the possible allylic bromides and then just double check to see if any of them are the same. Sometimes that happens. So this one has 1, 2, 3, 4, 5 unique allylic bromide products. And sometimes, I don't know, there might be a homework question that asks you, how many unique bromide products are there in this reaction? And if you don't understand that or look for that, you might get the wrong answer and be confused. So lots of complexity here. Well, I mean, lots of stuff to do, but don't view this as like a, you start at the start and you immediately get to products. You got to think through these various steps. And the workflow, I think, really should be, number 1, identify allylic bromide, allylic positions. Then number 2 is draw all the resonance structures that you can get from those allylic positions. And then step 3 is draw their resonance structures. And then step 4 is draw the bromides, convert them to bromides. And then step 5 is just look for any duplicates. So, lots of steps, but it's a way to tackle a complicated problem. You got to break it up into small pieces like this. So any questions about this one? And this isn't as intense as it gets. Most of the time it won't be quite this busy, but this prepares you for just about anything. All right. Let's talk about something different. Well, still radical based, but slightly different. We're going to talk about oxidation of lipids and antioxidants. So this is interesting because this gets to like some real world chemistry. These are things that happen, every day that affect like life outside of organic chemistry lab. So I'm going to tell you about some antioxidants. These react with reactive radicals to make them less reactive. And unable to undergo chain reactions. They're not going to destroy the radicals outright, but they're going to trap them into a low energy form that prevents them from doing any damage. So here is an important antioxidant that you've probably eaten recently. At least that would be my guess if you've had any kind of processed food, and most of us do. American diet's full of it. Just can't get around it. This one is called, for short, it has a name of BHT. So new thing to do when you're looking at when you're eating some kind of food, turn the label over and look for chemicals in there that you can start to recognize. BHT is one of the ones you'll see probably towards the end of the ingredients list quite a bit. It stands for butylated hydroxytoluene, or BHT for short. Butylated because it's got a couple of tert butyl groups on it. Hydroxy because it's got an OH. And this methyl group comes from the toluene part of this, and we'll learn about this nomenclature when we get to aromatic rings, but this is an antioxidant. So the way that this works is that if you have a reactive radical, I'm going to call this R dot. So imagine R dot is a reactive radical. Like it could be a Cl dot, could be an O dot. That's most often what it is. Could be a carbon radical, but some kind of reactive radical that's participating in chain reactions and causing lots of problems. Well, what can happen is that R dot can grab an H dot out of the BHT. And then you get a molecule that looks like this, where there's an O dot, and I haven't drawn the lone pairs in on BHT. I probably will right here just so you can see the complete structure. I'll draw them in over here as well. So you get this, and you get RH. So this is technically a propagation reaction because you haven't destroyed a radical, but you've produced it. You've produced a radical in the place of the reactive one that's a lot less reactive. This thing is quite stable actually because it's got a lot of resonance structures. Well, let's go through and draw them. This O dot can be shared differently by pairing it up with one of the electrons in the ring to give a new resonance structure where the dot is delocalized onto one of the carbons of the ring. All right, let's draw the result of that arrow pushing. It's going to give us a structure that looks like this, where we've got a dot right here, an oxygen carbon double bond there. The tert butyl group on the left side is still there. There we go. That's not the only resonance structure, though. You can keep going, pushing this electron further and further around the ring. I can take it from here and push it in like this and put it over here in the position exactly across from the oxygen on the ring. Still not done yet, because I can keep pushing that electron around further I can take it from here, push it over and share it there. Now it's on the left side of the molecule. And if you want to be really complete about it, there's one more resonance structure you could draw where you put the dot in, push it this way, push it back out onto the oxygen. That's a unique resonance structure because the double bonds in the ring are staggered compared to what they were where we started over on the far left side of the board. So kind of crazy, huh? Lots of different resonance structures. Whenever you see this sort of thing, what it tells you is that radical, because it's so delocalized, it's really stable. What do we got here? Let's count them up. We got 1, 2, 3, 4, 5 resonance structures, all contributing to making this radical more stable. So that's why this is such a stable radical, because it has all these resonance structures and it's like not very reactive as a result. Check this out too. It isn't just that it's delocalized, but look at the position of the radicals, like on the carbon here. This is on a carbon, that's a tertiary carbon, that's flanked by a tert butyl group. This is in a tertiary position. This is tertiary position flanked by tert butyl group. So, I mean, and the oxygen is flanked by 2 tert butyl groups. So not only is it electronically stabilized by being delocalized, but it's also really sterically encumbered. It's like really bulky. So it's hard for other molecules to get to the dot. So that's why BHT is such a good antioxidant. And if you look at vitamin E, alpha tocopherol, the molecule of the day, it has very similar features. It's got an OH on a ring that's got a lot of steric strain. So, so anyway, that's how antioxidants work. And they protect against oxidation at allylic positions in fatty acids, like we talked about with the survey question. So that is one of the things that's really useful about molecules like BHT. Do you have any questions about antioxidants? Yeah. So yeah, okay, so let's talk about what the problems with fatty acids. This is a good topic to discuss. We haven't talked about this yet. The book does talk about it. I'm going to talk about it a little bit, explain what the problem is with fatty acids, and what an antioxidant might do for you to help. So fatty acid oxidation. So why would you want to put an antioxidant like BHT in your food anyway? I mean, it isn't just put there for taste, I can tell you that. Its function is to protect the food from oxygen. So here's the problem with a fat that's in food. So this one is, I'm going to draw a fatty acid, oleic acid. It's a little different than the one that we put on the board for the survey question, but it's a little simpler. It has 18 carbons. Starting here, there's 1, 2, 3, 4, 5, 6, 7, 8, 9. And then between carbons 9 and 10, there's a double bond. And then the 10th carbon, it goes on 10, 11, 12, 13, 14, 15, 16, 17, 18. If you've got olive oil at home, there's a lot of oleic acid in olive oil. So, and one thing about olive oil is you got to keep it sealed up. This isn't one where you can really leave it out for a long time. You got to keep air away from it because it's got these allylic positions here and here. And those are subject to oxidation. It's easy to make a radical there because it's resonance stabilized. So the problem is that we are living in a sea of oxygen, which is both important for our survival. We have to have oxygen to metabolize our food and drive our metabolic processes, but it's a diradical. So oxygen O2, you can write it a couple of different ways, but this is the most common way to write it. This is probably the one way that describes it best. It's like a diradical. So oxygen is a really reactive kind of species, and it does react with lots of different things, which is both good and bad. We need its reactivity to survive, but then it also can attack our sensitive molecules like this. So anyway, what can happen is an O dot can react with one of the H dots in this fatty acid. Let's see, and then that goes there. And what you end up getting, this is kind of small. I want to make this a little bit bigger and a little bit more clear. So the O dot grabs an electron off the H dot, and one of the electrons, the other electron in that bond, goes to that carbon. And these structures are big, so it gets to be a little cumbersome drawing them. But I'll show you the way I do it. I start at the carboxylic acid carbon, and that's 1. And then I'll go 2, 3, 4, 5, 6, 7, 8, 9. And then 10. And then I'll go down 10, 11, 12, 13, 14, 15, 16, 17, 18. And what we've got here is a dot there. That should be an H. So that's an allylic radical. There's radical character here. There's also radical character here because it's resonance stabilized. But this can react with another molecule of oxygen. So there can be an oxygen that adds here, and then you've got another radical. I'll just draw that out here. So we're having lots and lots of oxygen around, and carbon dot can react with O dot. And that gives a new radical that looks like this. There's carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, and then 10, 11, 12, 13, 14, 15, 16, 17, 18. And we've got a new O dot here. So it's still a radical. This can react with another allylic position of another oleic acid, pluck an H dot off from an allylic position, and that can produce the following final product. I guarantee, I promise. Carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Has an O, OH. So this is called a hydroperoxide. And this fatty acid is damaged goods. It is going to contribute to off flavors in food. Eventually, if enough of this happens, you're going to not want to eat this food. And this scheme is in the textbook too, by the way. It's like there's a nicer picture than what I drew. But the point is these allylic positions are really reactive. And it just kind of goes along with the allylic reactivity theme that we've been discussing. So you aren't going to have to reproduce this scheme on an exam. I mean, I know it's just too much. But if you saw the scheme, hopefully you could follow the arrows. I think that would be a fair, a fair thing to ask. So just kind of study it and see. Hopefully it makes some sense. Okay. Any questions about this? Okay. So antioxidants work by intercepting any one of these reactive radicals, and the process just kind of keeps going over and over again. But if you had something like BHT or vitamin E, at any point in this mechanism, that antioxidant could intervene and provide an H dot here, and then you you'd stop the process. Or it could provide an H dot here, and you'd still have a hydrogen peroxide, but at least that reaction wouldn't progress anymore. You wouldn't get more propagation reactions. So, so that's the purpose of antioxidants, is to control this kind of process. And we have problems with oxidation in our bodies too. So there's naturally occurring antioxidants that are present in your cells that are meant to sequester reactive oxygen radicals that can cause problems if they're where they're not supposed to be. Because radicals are not controlled, they're not really controllable, they're really kind of indiscriminate in where they react. If a radical gets loose and starts causing chain reactions outside of where metabolism is supposed to happen, then you can have all kinds of cellular damage. So antioxidants are important for preventing that sort of thing from happening. And there's endogenous, naturally occurring antioxidants that go to prevent that. We can talk about those sometimes if you're interested. But yeah, same concept though. Sequester a reactive radical, make it less reactive. All right. And now for something really different. It's not completely different because it's radical based, but it's not quite the same as the other things we've been talking about. We're going to talk now about radical polymerization. And polymers, which polymer is a general term. It means, poly means many and mer means reactants, I guess. So it's a molecule that is made out of many individual subunits. Think of polymers as being kind of like molecules that are built like beads on a string. And what we're going to look at in this section is how alkenes can polymerize. So here is a scheme of what can happen and how these polymers, these plastics get made. They all start from alkene monomers. So the monomer is the starting material that's gonna go form the big, long molecule. Think of these as the beads that haven't yet been put on a string. Then if there is and you have to put an initiator in with these. Initiator, this is a molecule that produces radicals. And we'll talk about what initiators are in there, but these form radicals where there weren't any before. What will come out of this is a polymer. So a polymer is a molecule where we've linked many of these monomers together. And this is one way to draw it. An example of one strategy for drawing it. The squiggle means that's an attachment point. It just means that this is like the molecule keeps going in both directions. It keeps going to the left and it goes to the right. And it's just really big. And this is the skeleton, this is the backbone of the molecule. And the things that are on the chain are alternating CH2s and RHs. And if you're trying to keep track of where do these things come from, you can look for the smallest repeating unit. And what you'll find is that there's a repeating unit here. That came from one monomer. There's another repeating unit that came from another monomer. Here's another repeating unit that came from another monomer. And it keeps going and going and going. And these polymers tend to be very, very large molecules. They can have molecular weights that are in the thousands or tens of thousands, or even more. So they get really, really big. And one of the ways that people represent these, rather than trying to draw the repeating unit many, many, many times, a shortcut nomenclature type system that is really efficient at drawing a polymer is like this. So most often you'll see them represented with this kind of a structure where the monomer unit is put into brackets. This is the smallest repeating unit. And then the bracket, there's a set of brackets that are bisecting these bonds, and then there'll be a letter N down here. So N represents how many times this repeats. And N can be hundreds or thousands even. So these can just get to be really enormous molecules. So that's polymerization. And the difference between all of the different plastics we're going to look at, these are plastics that are important in packaging and in consumer products. They differ by their R group. So the R group is can be many different things, but it gives the polymer different properties. We're going to look at probably about 6 of the most common ones, maybe like 5 of the really common polymers that are present in consumer products. So, but let's figure out like how this works. What is the mechanism for the polymerization? How does it go from monomer to polymer? And it ends up being a radical process. It's a radical polymerization reaction. So let's talk about initiation first, because that's the first thing that happens in these processes. So step 1 is initiation. Just like we had when we were doing halogenation, chlorination, bromination of alkanes, you had to get some radicals to start with. And the initiators that get used in polymerization chemistry, there's a few different examples, but one of the really common ones that I'm going to show is benzoyl peroxide. I used to work in a polymer lab. I was in a group that did work on dental fillings. So polymers that go in your mouth and get used as restorative materials in dentistry. And this was one of the initiators that we used. It's called benzoyl peroxide. It's also present in over-the-counter anti-acne medications of different types. But it's got this weak oxygen-oxygen single bond. And when you heat that up, you can break that bond homolytically. And it generates 2 benzoyl radicals. You have to be careful with this molecule if you're working with it. Benzoyl peroxide is explosive. So this was one of the coolest demos I'd ever seen was a fire prevention demo, and it was done by the fire department, and they had like a bag of this stuff. It was maybe like a half a pound or something. They put it on a hot plate basically, and basically just everybody stood back and got hotter and hotter and hotter, and eventually it just exploded. It just went boom, and it was dramatic. So peroxides tend to do that. So if you ever have a molecule that has the oxygen-oxygen single bond in it, best to be careful with it. But this is a reactive radical. So this is an initiation reaction. We've got radicals now. Things are going to start to happen. So we're going to go back to our monomer now and see what it does. So when the monomer comes into contact with this initiator, I'm going to draw the initiator over here. The way that it works with the polymerization reaction is that the O dot reacts with one of the electrons in the pi bond to form a new oxygen-carbon single bond. The way you can show that is you could start a fishhook arrow at the dot on the initiator radical and show it pairing up with one of the dots on the alkene, and then the other electron in the alkene pi bond goes to that carbon. So this is technically a propagation reaction because we're not using up any radicals. We're just making a new bond, and we're getting a new radical out of this. All right, well, that's a new radical. It can continue. Yes?
Is this still like part of step one?
No, sorry, these are propagation reactions. So maybe what I should say here is I'm going to put, after the initiation reaction happens, then this is a propagation reaction. And all of these steps where the polymer grows are propagation steps because the polymer is getting one monomer unit longer with each reaction, but it isn't consuming radicals. You're generating a new radical in each case. So here we started off with an O radical. It reacted with an alkene, and we got a carbon radical. So we've not created or destroyed any radicals. We've just got a new radical. And this reaction can continue because there are going to be other molecules of monomer in there. And when this radical reacts with the pi bond again, it's going to generate a new radical where the chain gets one monomer unit longer. So the C dot here is going to pair up with one of the electrons in the pi bond. We're going to make a new carbon-carbon double bond here, or single bond here. And then that dot goes there. And with each propagation reaction, the chain gets one monomer unit longer. So now we've got a polymer that's grown one monomer unit longer, and it looks like this. We've got another CH2, CHR, with the dot at the end of the chain. And basically, this can just keep going over and over again. And it keeps going until two chain monomers, till two radicals find each other in solution and a termination reaction happens. So that could be any growing chain with a dot on the end finds another growing chain with a dot on the end, and they come together and the reaction stops. Or that carbon dot could find an initiator dot, react with it. Just make sure I'm this on screen here. So this carbon dot could react with this oxygen dot, and then the end would get capped up as with an initiator end. So in the end, what you get is you get the polymer with this bit in the middle, and the ends are where the initiator ended up reacting. So there would be an O initiator molecule here on the one side and an initiator molecule over here, the group that came from the initiator molecule on the other side. And if the polymer is small, if this N is just a few, like 1 or 2 or 10 or something, the N groups probably have a really important role in determining the properties. But if this gets to be like 1,000, then the N groups are pretty small and they don't really influence the properties of the thing very much. So mostly polymers, a lot of times people don't even draw the N groups, because by the time you make this really, really large, the N groups are kind of insignificant in terms of the overall properties. The larger the N gets, the different properties the polymer assumes. So polymers have kind of some remarkable properties due to being just so large in molecular weight. And it's what makes them unique in terms of their behaviors and makes them really good as materials. So I'm going to go over just a few examples of plastics that are made from free radical polymerization of alkenes. And it's some of the most important ones that you use all the time. So we're going to start with number 2. And so if you're ever curious about what is in your plastic bottle, one thing that you'll see if you turn over your shampoo bottle, or maybe if you've got a bottle of detergent like I have, brought some props today. I got a spray cleaner bottle here. This is got a number 2 on the bottom. It says HDPE. That stands for high density polyethylene. Polyethylene. And that's made from ethylene. So ethylene looks like this. Where the R group that we usually had there, that's just an H. So when this polymerizes, it forms polyethylene. And this one is really good for things like milk jugs, shampoo bottles, even grocery bags a lot of times are made out of HDPE. So next time you've got a plastic bottle of some sort, check the bottom of it. And we'll get to the disposable water bottles really soon. Those are a different one. That's number 1. That's PETE. It's a different class of polymer that isn't made by radical polymerization. So we're going to have to deal with those a different time. But for starters, we've got recycling simple number 2, which is high density polyethylene. That's the HDPE. That's where that comes from. There's a related polymer that's very similar in structure. It's got a number 4 recycling symbol with the circular set of arrows. It's called LDPE. This is called low density polyethylene. And it has the same basic structure as HDPE. It starts from the same monomer, but what makes it a little different is that every so often it has some branching. So instead of just one really long chain, sometimes the chain goes and branches a little bit. That gives it different properties. This one is more rigid. And this one is more flexible. And so it can be used for things that require kind of more of a stretchy application. Like for example, I brought in this, this is just a bag that's got some filter floss for Aquarius because I found it really fast, but it's got a number 4 and an LDPE in it. So inherently it's very similar structure to HDPE, but with just a little bit different physical properties. We're going to talk about some of the other polymers and finish this up when we come back on Wednesday. We'll talk about polypropylene, polyvinyl chloride, polystyrene. All right, see you next time.